Hi everyone, my name is Fatemeh from Concordia University, a PhD student from Concordia University. The title of my study is Modulatory Effect of Nicotinic Acetylcholine Receptors on the Human Memory T helper 1 and T helper 17 cells. At the beginning, I'm going to give you a general introduction on the, let me just bring highlighter. No, sorry. Yes. I'm going to give you a general introduction on the acetylcholine, which is the main neurotransmitter in the parasympathetic nervous system. It can target skeletal muscle, brain, and many other organs, so that following that, it can regulate the function of them, like changing the heart rate or the blood pressure. And in order to do its job, it needs to bind to its receptors. There are two kinds of receptors for the nicotinic receptors, muscarinic receptors and nicotinic receptors. And in this project, we're going to focus on the nicotinic receptors. Recently, studies have shown that immune cells can have their own cholinergic components, like they can produce their own acetylcholine and their own nicotinic receptors. Speaking of the immune cells, naive T helper cells and memory T helper cells are part of adaptive immune system. When antigen-presenting cells present antigen to naive T helper cells depends on the cytokines in the environment of the naive T helper cells, they can differentiate into different effector T helper cells, T helper 1, 2, 17, and Treg cells. In this study, our main focus are T helper 1 and T helper 17 cells. That T helper 7 and we're gonna consider interferon gamma as the representative of the T helper 1 as a cytokine that is mainly produced in the T helper 1, and T helper 17 for T helper 17, all 17A is the representative of the T helper 17. Regarding the memory T helper cells, there are different hypotheses about the formation of memory T helper cells. One of the hypotheses proposed that when the pathogen is cleared in the body, most of the effector T cells, like T helper 1, 2, 17, will undergo apoptosis, but the remaining will contribute to the memory T helper cells. We have different subsets of memory T helper cells, like effector memory cells or central. So in this study, we are considering the whole and general population of memory T helper cells. In this slide, you can see the combination of the immune cell, T helper cell, and cholinergic system, or acetylcholine and its receptors. As you see here, in the T helper cells, acetylcholine can be produced. So T helper cells can produce their own acetylcholine, and then acetylcholine can bind to its receptors, like nicotinic receptors. Nicotinic receptors are pentameric. They are ligand-gated ion channels, and they have like 1 to 5 subunits, and one of the main nicotinic acetylcholine receptors are alpha 7 nicotinic receptors, that all of those subunits are alpha 7. Eventually, acetylcholine binds to its receptors. The conformational changes would happen in the nicotinic receptors, and following that, the positively charged ions will move across the membrane, and depolarization would happen, and so some signaling pathway will be activated. For example, when nicotinic binds to its receptors, the JAK2 STAT3 pathway will be activated, and following that, it would inhibit the phosphorylation of the I-kappa B. When the I-kappa B is inhibited, is phosphorylation, so the recruitment of NF-kappa B inside of the nucleus will be prevented, and following that, the expression of the pro-inflammatory factors that were dependent on the NF-kappa B will be suppressed. Previous studies have shown that nicotinic receptors can regulate the function of the immune cells. For example, one of the studies showed that nicotine can lead to decrease in the level of IL-17 in T helper cells extracted from rheumatoid arthritis patients and some other studies. However, the role of nicotinic receptors, particularly alpha-7 receptors, in the nuclear signaling and regulation of cytokines in memory T helper cells is unknown. And also in this study, we want to investigate the involvement of nicotinic receptors in the regulation of memory T helper cells in terms of cytokine production. For the material and method, we obtained PBMCs and from healthy participants, and then we isolated memory T helper cells by negative selection magnetic bits, and then we expose memory T helper cells for 5 days with nicotine as nicotinic acetylcholine receptor agonist. Nicotine in this case can mimic the effect of acetylcholine by binding selectively to nicotinic acetylcholine receptors. And GTS21 was another agonist that selectively binds to alpha-7 nicotinic receptors. After 5 days incubation, we collected supernatants and we measured the cytokine on interferon gamma and IL-17 by ELISA, and we ran flow cytometry and Western blot to measure the intracellular cytokine and phospho-NF-kappa B expression. Regarding the results, at the beginning we started with the PBMCs, and we found out when we treated the PBMCs with the nicotine, the level of interferon gamma significantly reduced. So in comparison with the ACT group. The ACT group in this case are the cells that were exposed to anti-CD328. No ACT are the cells that were not exposed to anti-CD3 and 28 and nicotine. But the results of IL-17 showed that nicotine cannot change the level of IL-17 in the PBMC. So the result of interferon gamma was a hint for us to work on more purified cells like memory T helper cells. And surprisingly, in the memory T helper cells, we found that when we exposed them with the nicotine, the level of both interferon gamma and IL-17 significantly decreased after 5 days of the incubation. Then we did intracellular staining. So we panel A is showing non-activated cells, B is showing the activated cells with anti-CD328, and C panel is showing the activated cells with anti-CD328 and stimulated with nicotine. And as you see here, after treatment of the memory T helper cells with nicotine, the percentage of CD4 positive IL-17 T helper cells show a significant decrease in comparison with the ACT group. So these results were in line with the ELISA results. We did the same experiments for the interferon gamma, and as you see, the percentage of CD4 positive interferon gamma positive T cells after exposure with the nicotine showed a significant decrease. In the next step, in order to see which signaling pathway is involved in the visual effect, we measured the phosphorylation or phosphorylation of the NF-kappa B by Western blot. So we exposed memory T helper cells with nicotine, and after 15 minutes incubation, we measured the phosphorylation of phospho-NF-kappa B and total NF-kappa B. And as you see, based on these results, this ratio, phospho-NF-kappa B per NF-kappa B, after exposure of the memory with nicotine, there was no change. We had just a decreased trend because we expected that the nicotine can decrease the level of this ratio, but it didn't happen. So maybe some other signaling pathway are involved. So in our future plan is to measuring some other signaling pathway and transcription factor. And also we wanted to see during these 5 days of incubation of memory T helper cells with nicotine, if nicotine can change the proliferation or cell division. And as you see again, A is the panel that the cells were not activated, panel B are activated with CD328, and C the cells that receive nicotine with anti-CD3 and 28. And nicotine could not change the cell division within the 5 days incubation. For the next step, in order to narrow down the project, we decided to work on more like specific receptors. So we decided to particularly focus on alpha-7 nicotinic receptors. So we use GTS21 as an agonist for the nicotinic alpha-7 nicotinic receptors, and ELISA results showed that after exposure of the memory T helper cells with the GTS21, after 5 days, the level of interferon gamma and IL-17 significantly decreased in comparison with the ACT. In this plus vehicle or DMSO, they are the solvents of the GTS21, and there is no significant difference between the vehicle control condition and ACT condition. So in conclusion, activation of nicotinic receptors, particularly alpha-7 receptors, suppress the production of interferon gamma and IL-17. It was like suppression of TLPR1 and TLPR17 cells. And our findings suggest that targeting nicotinic receptors in memory TLPR cells can be an approach for manipulating immune responses to control inflammation in autoimmune diseases like MS. Since this project is an ongoing project, the next plan is to knock out alpha-7 nicotinic receptors gene to see what would happen in case of loss of function of this receptor, to make sure the visual effects is mediated with alpha-7 nicotinic receptors. And we would measure some other transcription factors, and we would evaluate some other signaling pathways that might be engaged with the effects of alpha-7 nicotinic receptors. And finally, we would work on the memory T helper cells of EAE mice model to see what would be the effects of nicotinic receptors in their pathological conditions. And finally, I'm going to acknowledge my supervisor, Dr. Darlington, and all of the lab members for your help and support, and BSI for this opportunity to have a talk today. Thank you so much. If you have any question.
There is a question in the chat. Were the doses of nicotine used for the different experiments the same, and were nicotine effects dose dependent?
That's a good question. And for all of the conditions, we use the same dose of nicotine. We didn't run experiments for the dose. We didn't use different dose of nicotine. And 10⁻⁻⁵ molar is the average amount of nicotine in the blood of cigarette smokers. So that's why we picked 10⁻⁻⁵. It's something between 10⁻⁻⁵ to 10⁻⁻⁶.
And another question, what are some of the other important nicotinic receptors on T cells? Do you think your results from the nicotine simulation are completely driven by A7 and AChR or other receptors as well?
Yeah, that's a good question. And that's why we're gonna knock out the receptors. The reason that we're gonna knock out the receptor is that we use some inhibitors like antagonists for alpha-7 and antagonists for the whole nicotinic receptors. But unfortunately, those antagonists did not work very well, or maybe this GTS21 is working well other receptors other than alpha-7 receptor. That's why the next step is knock out, like physically we're gonna knock out alpha-7 receptor and see what would happen.
And one more question, how does change in the microbiota impacts the nicotine effect on T helper cells?
That's a good question. I've never thought about it, but maybe I apply it for the next part of my experiments. I think about it and see what would be those effects on the nicotinic receptors, but absolutely they would have some effects.